am totally going to pivot. We've been talking about prevention and data and clinical trials. I'm going to talk to you guys about animal models because unfortunately I've been a drug developer for over 20 years and we still don't have good animal models. I've probably cured a bazillion mice with cancer. I put the cancer in them and then I cured them. But I've yet to actually cure that many people of cancer. So I'm gonna kind of walk you guys through a little bit about this ONCOPIG that we've built. So I'm preaching to the choir here when I say cancer is the leading cause of death, and we've really made huge progress with the immuno-oncolytic drugs. You know, we see the commercials of Keytruda and everything. As well as if you think about the super chemos, so antibody drug conjugates that get the payloads directly into the tumors. So we're making unbelievable strides, I would say. Still a long way to go with cancer therapeutics. But one of the things that we have not done well is building better animal models that are more translatable to the clinic. As I mentioned, I'm always doing a test in a mouse for efficacy, and then I look at my large animals for tox as well as metabolism. So the problem is mice are much smaller, right, than us humans. So they don't also metabolize drugs the same way as humans. Well, pigs are an ideal model system. We always use monkeys because we said monkeys are similar to us from a genetic standpoint. They're 99% similar to us. A pig is a 98. And we use pigs all the time in other forms of research, so cardiovascular, immunology, but we don't use them that often in cancer. So what did we do? So our co-founder and chief scientific officer, Larry Shook, actually was the leader back in 2012, led the entire mapping of the pig genome. And from this mapping of the pig genome, we were able to create our ONCOPIG. So what our ONCOPIG is, it's a genetically modified pig that we've actually put two common driver genes within this pig, P53 as well as KRAS. And we actually have a stop codon in front of it, and it's flanked by two proteins, basically. And what we're able to do is we inject a adenoviral vector into these pigs, whatever organ you want this tumor to be in, and it pulls that stop codon off and allows the tumor to be generated. So we're able to induce the tumors in any organ. We validated it in over 8 model, actually almost 15 model systems. And we're continuing to develop better and better models. So a little more on the specificity of how does this work. We have two main, I would say, model systems. We have what we call a non-specific tumor histology model. And so what that is we basically inject the vector into whatever tumor you want. So for example, if you wanted a liver tumor, you'll inject this AdCre solution into the liver and a tumor will form. If you want 4 lesions, we usually say inject 5 spots and you'll get 4 lesions. Now this is a non-specific model. This non-specific model meaning it's undifferentiated cancer. They're neoplastic cells, but they're undifferentiated. They're sarcomatoid based as well as whatever organ that you played in. So there could be some adenocarcinoma of the hepatocytes. Then we have what we call a defined histological system. Now what this defined histological system is we actually have those liver ONCOPIG cells. We inject those liver cells, and then we take those cells back into the animal. So that's very specific. So if you wanted an adenocarcinoma of the liver, we could do that. If you wanted an adenocarcinoma of the lung, we could do that as well. The other thing we can do is we have CRISPR technologies that we've in-licensed, and if you want a specific protein overexpressed as well in your tumors, we can do that. If you want a specific point mutation within that protein, we could do that. So this is an ideal model system if you think about it, because you can test all three questions: efficacy, safety, and metabolism of your drug within this model system. So what have we done thus far? We have multiple different cancer tumor models, as you can see here, in a wide variety of diseases. The couple of things that I can tell you about the model is we have 100% tumor success rate. This is very different if you've ever looked at genetically modified animals. And we are able to induce these tumors within 1 to 2 weeks. We have full tumor formation formed within these animals. So now, this is the reason I always talk about this being an ideal. Again, I don't need 2 different animals, a small animal and a large animal, to ask all my questions. I have one animal model system that I can ask everything, and it's an animal model system that's as close to a human as we can kind of get without actually testing that question in the human. So this is a great tool for researchers. As you can see in the images here, when we have the induced tumors in the pigs versus the human, there's really no difference. As well, you can see in different visualization tools, you can see the angiogram at the bottom as well as the CT scan there. And then what's really cool, as I mentioned, not just thinking about you can ask all these 3 questions, but it's a large animal. So now I can collect all of that tissue that I need. Someone was talking about why doing biopsies on the patient. Yeah, I mean, it's a lot of 5 days off work to get all the, but I have a pig now. You can take all the biopsies in the world you want with your pig. You can take all the blood that you need from the pig, because of course this is a large animal, much different than when we tested all of this work within mice. Oh, wrong way. Now, let me go over a couple use case examples, because, you know, I can talk about our pigs all the time. But this is actually with one of our researchers. They wanted to actually test a immuno-oncolytic within a liver tumor. And what you can see here is on your far left, you see the liver mass. This is after we've induced the pigs. We had a liver mass, and you can see it's about a 22 millimeter. What they ended up doing is they were testing 2 questions. They wanted to be able to specifically show that they could locally deliver that medicine directly into the liver, and that's where you can see the catheter-directed delivery. So they wanted to be able to show that they can get the drug into the liver, but then they wanted to also be able to show, does the drug work? Because this was a new immuno-oncolytic based drug. And what you can see here from the left to all the way over to the end, you can see they were able to show about a 50% reduction. So we do targeted targeted based therapies. This is a different example. This is a device. This was looking at a new tool for electroporation. This particular customer, what they wanted to do is they wanted it in colon cancer. You can see clearly before treatment the mass within the colon, and then after they did the electroporation, a complete ablation of the tumor. So I talked a lot about what our ONCOPIG and what we could do. The other thing that we are building upon is the technologies, not only to be able to have that specific tumor, but now we're editing within an overexpression of a gene or an overexpression of a particular point mutation, because as we've all heard in cancer or drug development, we're not just doing chemotherapies, which will just, take any type of cell. We're now going down to a point mutation. So whether it's an overexpressed, like a HER2, We talk about HER2 therapies, if it's a HER2 expressing tumor. Well, we're even able to not just look at like an overexpression. And I think David was talking yesterday in the morning about the KRAS and inhibitor drugs. These are now not just broad KRAS inhibitors, they're now going down to single point mutation directly for that. And so we're able to actually build within our pig models a specific model down to that point mutation. So this just really talks about why is this a much better platform than what we've typically utilized. We talked a little bit about the genetics. This is the most common or just as common genetically as a monkey. The other thing we didn't talk about is this is a system, it's pigs. The ethical conundrums that we have when doing dog studies or monkey studies, we don't have that anymore. The issue that we have in sourcing monkeys now post-COVID has been unbelievable. And for those of you who know this space, monkeys are unbelievably costly. Pigs are not. And so this is, again, an awesome tool that really we are hoping more people will utilize this, because ultimately the goal is to reduce cost, time, and risk before you get into the clinic. So I'll leave with this. It's a model that you could use with anything, whether you're doing, we talked about drug testing, you can also do maybe a new imaging tool if you wanted to understand more. We can collect additional tissues if you're trying to develop a diagnostic within the space, or better biomarkers. So we're super excited about this. I will tell you that the company's been around for about 4 years, and I've come on to lead them in the past 6 months. And we've really started to generate, and we're training almost every CRO out there. So for those of you ever wanting to utilize a pig to get your drug or your device into the clinic quicker, I'm more than happy to collaborate with you to move forward. So I will end with that.
Thank you for the presentation. This is really an exciting alternative, actually, for the current models. So I'm just wondering, like, in order to have the investigators interested in this model, what's the cost difference between the transgenic mouse model and then your ONCOPIG? Just some perspective.
Some perspective, it's about double to a mouse,
Okay.
but it's about a fifth of a monkey.
Okay.
So, and again, I always try to tell individuals, because everyone's like, oh, a mouse, it's so much cheaper, and I can do 20 of them, right?
Right.
Well, if you think about it, your drug is going to work in a mouse. It always works in a mouse. I can tell you a lot of research. You're more looking at the best you have as a mouse model. And the transgenic mouse models are great. That's all we have right now, other than this. And again, we have limitations even within this model. I don't have every organ system up, because some organs for the pig are not ideal to create cancer.
Yeah, in terms of reagents, so it's like for mouse reagents, like a lot of them, they're really well developed, so it's like readily available. But for the pig, so it's like, can you comment on that?
Oh, yeah, sure. So our pig particular, so we've gone through the process to get it cleared by the FDA. And what that means is we have been able to prove to the FDA that we can sell this anywhere in the United States. We're actually also going abroad. We actually just partnered with a CRO, and I literally flew pigs to Spain. So pigs can fly. I really did it. And we have our first population that we're beginning. So basically what you have to do to clear these animals is you have to be able to prove to the FDA that the transgene is in every single piglet. And then that, because they're genetically modified, there's no risk for them to get in the food chain. And so we were able to do that. So if you wanted to purchase a pig, you just call us up and we can whatever, send it wherever you would like in the United States.
I have two questions. One is, and it's a challenge we have with, because mice are used and pigs. You listed breast cancer. Breast cancer, 90% of it's postmenopausal. Pigs don't go through menopause, mice don't go through menopause. So you're not actually looking at the natural course of the disease. You're looking at a xenograft of the tumor, not, so I guess part of the question is, does the pig respond differently than the mouse in that sense? And one of the jokes everyone makes is, we've cured cancer in mice, but they don't have insurance, so it's not really a profitable business. Yeah.
Yeah, great question. So as I said earlier, there are model systems that are very good and translatable into the clinic. Breast is not from this pig.
Okay.
Where our breast model works well is if you're testing a device or you want to be able to surgically handle and ensure.
Sure.
But it's not good from a translatable, that's
Okay.
why like liver or lung
Yeah.
model is definitely more translatable.
Okay, and the second question, on the table where you were comparing different animal models, you mentioned, or you listed that you can look at comorbidities.
Yes.
And the average person has 5 comorbid conditions besides the cancer that they present. How do you induce those? And
Yeah, great question, and I didn't speak about it. So we have 3 comorbidity models that we've developed. So we have a cirrhotic liver model. You know, you'll laugh, but we've literally have a protocol to give alcohol-induced cirrhotic liver disease. So when we say if you want to not just have liver cancer, but you want liver cancer that's associated with a cirrhotic liver, we have that comorbidity model. We also have a diabetic model because pigs are really good from a diabetic model as well. So, and we're also building a miniature version, which the best way to describe the miniature version is currently our pigs are very big. They have to be in a facility like as a non-human primate. Our miniature version will really, I think, open up the opportunity because it's about the same size as a dog. So we'll have that next year.
any
I
other
have a
questions?
question, maybe a couple of them. So are these like appropriate for proof of concept, or have they been used in IND enabling studies? I know you said you have FDA clearance. Have they been used in any IND submissions?
So the FDA clearance is to be able to sell the pig to ensure that the risk isn't within. We have actually worked with device companies to be able to get our pigs utilized for the 501s, but as well as that liver study was an IND enabling study. Now, that IND isn't approved yet. They have some CMC issues. So again, as you know, with INDs, everything has to come together, not just, but the FDA has seen it, and they had a couple questions with regards to the pig model for that particular study, and now they're just waiting for their CMC clearance.
And how do you address the genetically modified organization question? Obviously, that's a big question for the European agencies.
So we have to show that we're able to have it in a controlled environment, as well as that you cannot introduce it into the food chain. And if it were to get introduced, that there is no deleterious effects within the animals. So in Europe, genetically modified animals are becoming the huge shift because the ability to get non-human primates is becoming extremely challenging, and the European guidelines are really pushing away from the use of large animals such as monkeys.
Let's thank Jessica again. And I'd like to present you with a certificate for your presentation.